Leading a startup team, whether you're delivering a sugar rush, stocking coffee, or getting a regular delivery of snacks, Office Depot has solutions that fit every startup culture, from getting those first business cards and stationery to ordering fleece pullovers with your new logo. To learn how Office Depot and the California Technology Council have partnered to bring you savings on all of these startup essentials and more, go to californiatechnology.org/member benefits. I'm Daniel Levine, and this is the Bio Report. Nurix Therapeutics is harnessing the body's natural process for controlling protein levels to target and degrade proteins that drive cancer and other diseases. Its small molecule therapies control key enzymes responsible for protein breakdown and can be used to modulate the levels of proteins within cells. The company believes it can leverage the approach to treat a range of diseases. We spoke to Arthur Sands, CEO of Nurix, about the company's discovery platform, its pipeline of cancer therapies, and its recent collaboration with Gilead Sciences to discover and develop drug candidates against up to 5 targets. Arthur, thanks for joining us.
Thank you for having me.
We're going to talk about Nurix Therapeutics, its platform technology for targeting harmful proteins, and its partnership with Gilead Sciences to discover and develop up to 5 disease targets. Perhaps we can begin with some basics. What role do protein mutations or dysregulation play in various diseases?
Yeah, so these mutated proteins are generally the drivers in cancer. That means they basically cause cells to divide in an uncontrolled way, which leads to a tumor. And so proteins that are mutated are obviously bad actors for the cell, and so we basically are able to take them out. That is, to actually degrade them or destroy them and block, therefore, the proliferation of cancer cells. That's the goal.
There are normal cellular mechanisms for controlling proteins that Nurix is trying to harness. What are these and how do they work?
So normally the cell is able to control or regulate its proteins, and that's how it controls its life cycle and all of its metabolism. But there is a machinery in the cell in order to accomplish that. That machinery is known as the ubiquitin proteasome system, kind of a complicated name, but the term ubiquitin originally was used because these proteins these control mechanisms were found everywhere. They're ubiquitous. And the cell can tag proteins for degradation or for destruction as a mechanism to regulate them, to control them. And many of the proteins control cell division and cell replication. And if one can harness that, which we can with our drug molecules, one can actually redirect the cell or basically give it the incentive via these molecules to destroy harmful proteins to the cell. And that's really what we do, and that's what Gilead is interested in, is being able to make new drugs that can redirect this natural protein homeostasis. That's another term that's used, so this protein balance for the cell, such that you can treat disease.
Why does being able to degrade or modulate the level of a protein represent an attractive therapeutic approach?
It's a new approach, number one. So new approaches are always welcome for intractable diseases where we really haven't done very well, like cancer. It's a new approach, and it's one that uses, again, the natural, cellular machinery, the natural body's natural machinery to actually regulate protein levels. But it's more than just being new. It's actually catalytic, and what that means is once you harness it and turn it on, it amplifies its effect. It amplifies kind of naturally its effect. So you can get an amplification of the therapeutic effect within the cell with this type of molecule. And normal drugs or regular drugs that we've had for decades don't do that. They basically inhibit a particular target or block it for a period of time, and then they wear off. These drugs have an ability to persist, if you will, and use this catalytic mechanism in order to exert their therapeutic effect.
People still talk about so-called undruggable targets. How does your approach provide the potential to target proteins that may have been out of reach to other therapeutic modalities?
That's a very good question and a very big point. So the traditional drugs need to block a particular protein via an active site or a catalytic, a site like an enzyme site or some other particular mechanistic site of the protein. Many proteins don't have that, and so before this technology, they were unapproachable for drug discovery. So this approach allows us to basically tackle any protein, whether or not it has an active site or some kind of enzymatic site. We can tackle it, and we can knock it out or degrade it. So that's the big difference. And there's a whole really host of proteins that have been known for a long time in cancer to be mutated and deregulated and be the causes of cancer that we, by traditional drug development, we've not been able to tackle. We've not been able to hit them. This offers the approach, the promise of being able to degrade those proteins.
You're not alone in pursuing protein degradation as the basis for therapies. What's different about Nurix's approach, and is there a case to make for why what you're doing is better?
Well, indeed there is. We think there is, and I think Gilead does too. So in order to trigger this mechanism to degrade proteins, one has to harness a particular enzyme within the cell, which is called the E3 ligase. So there are, in the genome, in our genes, there are over 600 E3 ligases encoded in our genome, which is a very large number. And what Nurix has done is been able to really harness many of these ligases. So the entire field has been working really on 2 E3 ligases in general. And right now, Nurix, we've screened over 20 of these ligases. And we've built the chemical libraries in order to do that. So in order to harness an E3 ligase, one needs the chemical matter to actually be able to build a drug. You need the molecules. And we have invested heavily in a particular type of library technology, which is called DNA-encoded libraries. It's sort of an interesting and very powerful technology, but it's where you use DNA to actually tag and code small molecule libraries that allows you to hit or bind to really unique proteins. And these E3 ligases are very unique proteins, and so you need the libraries of chemical molecules in order to do that. So we built that, and in fact, they're working quite well, and we are finding new chemicals that actually will interact with these ligases, which allows us to build these harness molecules and actually harness this machinery to then degrade target proteins.
You've got two lead preclinical programs. I thought we could walk through each of those. The first target's BTK. What is BTK and what indications are you expected to target with this?
So BTK is a protein that will, when degraded, will allow you to treat lymphomas and leukemias. So it's a very important control protein for white blood cells, that is lymphocytes, within the body, which if they grow out of control, you basically end up with leukemia and lymphoma. You have a proliferation of these cells, and it's a blood-borne tumor that is, of course, fatal. So BTK, which stands for Bruton's tyrosine kinase, is a particular protein that controls a particular B-cell lymphocyte development. So a white blood cell, which is a very common form of leukemia and lymphoma. So BTK needs to be degraded in order to treat lymphoma and leukemia. There are drugs out there right now that inhibit BTK's activity. They sort of block it for a while. But there are mutations arising in BTK that make it resistant to the current therapies. And so our approach is to basically take out BTK, to destroy it, and hopefully resistance will be futile.
Is the expectation that this would be a standalone therapy, or would you expect to use it in combination with other therapies?
Everything we've seen so far in our mouse models of lymphoma tells us it can be a standalone therapy. Now, but however, in addition to that, it is important to develop it with regard to combinations, because patients are often on, have been on usually multiple therapies in order to try to treat their disease. So we are looking at combination approaches, but we do see single agent activity in all of the preclinical models of lymphoma.
You're also pursuing CBL-B as a target. What is that, and what indications do you expect to pursue there?
So CBL-B is a very interesting E3 ligase. So that is one of these ligase proteins I was referring to earlier. And it is a key control point for the immune system itself. So in this case, we're not trying to necessarily, CBL-B is not a target for cancer. It is a target that if we hit it, we can stimulate the immune system. So normally CBL-B downregulates or suppresses your immune system. And in the case of cancer, we want to turn on the immune system so it can see the cancer, see the tumor, and actually attack it. And this is the whole field of immunotherapy. So CBL-B is a very interesting molecule. It's an E3 ligase. No one's been able to drug it so far, make a drug to this thing. We think we've got it. It is an oral agent, and it is a very powerful stimulant of the immune system.
And would that be used in combination with other immunotherapies or by itself?
Well, we're going to find that out. We think it can be used by itself. Again, we test everything, first as single agent. However, again, it's cancer is a polypharmacy sort of approach, right? It's a war. So we do also want to be able to see it in combination with other immunotherapies that exist today. And we think it will actually be very versatile in its ability to combine with current therapies.
In June, you announced a broad partnership with Gilead Sciences that involved a $45 million upfront and potential milestones of $2.3 billion. This covers developing therapies for up to 5 targets. Is there any target you're pursuing yet as part of that collaboration?
Well, we've already initiated all 5, and of course, we have not disclosed exactly what those targets are. and we're moving on them. Some of them are, I guess I would say, some of the better known targets in cancer in the scientific community, but to date they have not been druggable, and so these are proteins that everyone would love to be able to hit, and I think we're going to have a really good shot at it.
Any way you're going about prioritizing indications you'll be pursuing?
These are the type of cancer targets that basically will allow us to hit many different tumor types across, from pancreatic to colon to breast cancer. These are a very broad, potentially acting drug targets, and that's what makes them very attractive.
And is Nurix expected to play any role in the development of these candidates?
We have the ability to actually develop these candidates in collaboration with Gilead for 2 of the 5 programs. That's a very important business point, is that we can participate in a 50-50 cost-profit share with Gilead for the U.S. Development of these oncology future products. And that's going to be a real value driver for Nurix, because it's very important for an emerging biotech company to own a substantial portion of the product rights for its future drugs.
One of the challenges of collaborations like this for a company like Nurix is that you have to place a fair bit of faith in your partner. What confidence do you have that Gilead will prioritize these programs and move with the speed you'd like?
Well, Gilead has an absolutely superb chemistry team that has developed a number of blockbuster drugs. They basically changed the whole medical rule book for hepatitis C with their breakthrough drugs. It's really amazing what Gilead has done. And they understand what it takes to actually make a breakthrough agent. And so their chemistry team has been here. We're both here in the Bay Area in San Francisco. We have 5 teams already on the 5 targets, working very closely together. And we believe, and we know actually by working with them, that they're going to be a very powerful partner for Nurix.
Arthur Sands, CEO of Nurix. Arthur, thanks so much for your time today.
Thank you for having me.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send an email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.